back carpal tunnel, but the point is I have lots of experience, and right now I'm standing before you as scientist John. There is John, the business person, but right now I'm going to even talk about competitors, and right now all your questions will be answered as directly as we can. So about Protify, we were founded in 2014. I did my postdoc at Cold Spring Harbor Laboratory, and CSHL is an amazing place. If you have an opportunity, you should go. We've now moved to upstate New York, where we're expanding facilities, and I literally left construction of that to come here. I founded Protify in the laboratory of Dr. Daryl Papin. Daryl is one of the 4 founders of proteomics, and there are truly 4. There's John Yates, American, Matthias Mann, German, Rudy Ebersol, Swiss, and Daryl Papin, who's British. Those 4 people, over the past 20, 30 years, have really built the foundational pieces. Those people have built the foundation that we work on right now, and that includes everything from reagents like isobaric labeling, Daryl invented that, to the software that we use to search. And so we are problem solvers. I didn't, as I said, form a company because necessarily I wanted to. I formed it because I needed to. And what's the goal? You guys know that? You guys are younger, so this is now dating me. That device in the upper right-hand corner is a tricolor, and it would scan the fictitious person and tell the fictitious doctor in the science fiction show everything that was wrong about the person. And ever since I was small, like really small, I wanted to make the tricolor, and that desire guided my whole career. So in proteomics, we start with a beautiful sample, which is well organized, and we make our lives much, much more complicated. We first destroy it by lysis, which you're going to do. Then we destroy it further by, in bottom of proteomics, doing proteolysis on the samples. And then with a lot of bioinformatics work, we try to reassemble the picture. Fundamentally, sample prep exists because the properties of proteins are very diverse, and they're not completely compatible with the systems that we use to analyze proteins, at least in mass spectrometry. There are new technologies like Nautilus Bio. They are not subject to the same physical principles of measurement, and that enables it. However, if you have a full-length protein and you try to fragment it, no matter how you fragment it, unless you have a lot of knowledge about what that protein is, you're not going to be able to understand what those pieces are. And the properties, physiochemical properties of proteins are very diverse. So blood is water soluble, mostly. Brain is lipid soluble, mostly. So how do you come up with a single protocol to handle that huge range of diversity? The answer is we turn it into peptides. Now, sample prep is a massive field, and people don't really appreciate how important it is. But remember, if you have a substance in your sample that is a contaminant or prevents chromatography or ruins chromatography, you have biological information that you're unable to access because you haven't paid attention to biochemical realities. And that's things like lipids. Lipids are very interesting, but we use a whole different set of protocols to analyze lipids than we do for peptides. That's a simple example. And lipids are a frequent contamination, especially phospholipids, if you're working with blood. That's a single example. DNA is long and stringy. Polysaccharides can be slimy. Insoluble material will clog columns. That's probably one of the biggest challenges that is, I think, the biggest challenge in nano LC. A little speck of dust will cause your samples to fail. And of course, we have an analytical device called a mass spectrometer, which is sensitive to salts, detergents, polymers, urea, all of the stuff that we typically use on a benchtop. Those are things that you have to worry about when you run it into a mass spectrometer. How much should you care? Well, that's why there's this graph. This is my friend Paul Pahowski at Pacific Northwest National Laboratory. And this is the source of variability in bottom of proteomics for one lab, one technician, and one other scientist. Sample preparation, if you include digestion, is a total of 75% of the overall variability. And that's with the same person's hands. Now imagine you take 2 or 3 or 4 people or different laboratories with different protocols, different ways to solubilize things. This becomes then more like 100%. So sample preparation really, really matters. At the very beginning, you have to ask yourself, if there's something I need to remove, or is there something special? Serum has a very large dynamic range. Albumin is 90% of the protein, or something like that. How do you lyse? Did you lyse? Different samplings are very, very different. So lysing microbacteria or yeast is very, very different than lysing HeLa cells. And if you use the same protocol that you've used on HeLa cells, you will probably get very bad answers. The typical workflow is pretty simple. Simple, right? You have some kind of sample prep. You have some kind of analysis. And this means actually running it in an instrument. Perfect, thank you. And then you do some kind of bioinformatics. And what I just said is some kind of, which means we have a really big problem. Fundamentally, there's a lot of steps, no harmonization, and no ground truth. Ideally, we would measure biology, and then that would be our ground truth. But because of all of these different steps, mostly in the sample front area, that's the graph I just showed, we don't end up getting the same truth. And this is what I just said about blood versus brain. And there are different ways to digest things. And now there are technologies, specifically Nautilus, that doesn't require digestion at all. I want to ask you guys, how many people here have worked in the laboratory? Raise your hand so I can see you. It's like 2/3 approximately. Looks like that. So how many of you have done a lysis of some sort? All right, and after you lyse something, what's the next step? Yeah, centrifugation, right? The lysate was clarified by centrifugation. And what do you do with that? Typically, you throw out the pellet. But my bones are important, and if you put a bone in water, it doesn't go into solution. And my muscles are important, and if you put muscle in water, it doesn't dissolve. So do not forget that just because something is not soluble in the condition you chose, does not in any case mean it's not important. In fact, many, many things are extremely important. And don't forget, everything in life passes through a membrane. Information, food, waste, everything, water, it all goes through a membrane, which is obligatorily hydrophobic. So by throwing out all this stuff that's just not soluble, you're missing very large pieces of life. As a personal example, I did protein lipidation, and I wanted to analyze the pellet. Why? Because if you put a lipid on a protein, it probably decreases in solubility. And when I did that, I found out the histones are lipidated. That is an entirely new class of PTM histones. And it has yet to be researched in case anyone is interested, to my knowledge, in a project. Okay, this is text heavy, but you guys just need to think, and I'll go through this. Before you even get started, was the experiment properly designed? If you do end up working in a laboratory that has a mass spectrometer, almost certainly you will have collaborators because the mass spectrometer is very expensive. So people will come to that one mass spectrometer or 2 or 3 or the core or whatever. And they'll have something, and often they have something that they don't really know what it is, and they don't know biochemically what's in the container, and they don't know how much there is. So you have to start with the basics. Was the experiment properly designed? Meaning, if the experiment is botched, don't bother analyzing it. What's wrong with just 1 plus and 1 minus? Let's say I give you 2 averages, and I want you to tell me if they're different. Can you tell me if those are different? Anybody got an idea? Let's assume they're from a Gaussian distribution and you're going to use a t-test. What do you need to know to tell me how different they are statistically? Say again? I couldn't hear you. Yes, of course, but you can count that. Oh. What else do you need? Standard deviation, right? The problem is, if you have one minus and one plus. You have no idea how broad those curves are. They could be these really tight curves, so that a very small difference is significant. They could be these huge broad curves. So you must have replicates. Now, there are instances where people will cheat this. I don't think it's worth cheating, especially because analytical times have gotten faster.
Sorry.
They can't hear? Is it this?
Uh,
Where's the audio?
that is.
No, where is the speaker for the audio of the next room?
It's in that room. We have kept it at full.
I understand. So is it this speaker that's communicating the audio to the next room?
The mic is communicating.
This is.
Yeah.
Can you turn this down so I can talk louder? Meaning turn the volume down because I would yell otherwise. Yeah, there we go. A little more. Okay. All right. So I'm supposed to speak louder. I can do that. Now, how was the sample collected? How was it stored? How many people have faced a freezer failure? When I built my lab, one of the first things I did is go buy a generator. And it's a problem, right? So did something happen? And